Hello, I'm David Weisblad. I'm a professor in Cell and Developmental Biology Division within the Department of Molecular and Cell Biology. And in my lab, we study the development and nervous system and behavior of leeches. How do you get from a single embryo to something that looks like a leech? And what would you do? Well, you start out by describing what you see. Okay? And in the leech, it turns out that there are stereotyped patterns of cell division, so that even when the cell makes when the embryo zygote makes its first division, you can tell the two cells apart in embryo after embryo because they're different in size and their cytoplasm is a little bit different.
Okay.
And then you can go on from. In fact, there's so many interesting things about the first division. You can spend your whole life studying that one cell division, and people have in other animals. But
wow, that's
right,
cool.
because it just the signaling processes. Why is it unequal? How does there's cytoplasmic rearrangements, stuff like that. And for every cell division, you could spend right. But we're going from one cell to 100,000 cells or something like that. So it really is many lifetimes.
Okay.
But you start looking, you see a pattern of what's happening, you start asking questions. Oh, this is weird. How does this, or you form a hypothesis about if this works. I'll be able to pour the blood in. And it will not leak out. So far, so good. So now you can see the leeches they know there's something there. They're not really super smart about finding it. But some of them have latched on. This one.
How do they detect it? Do they have like olfaction?
Yeah,
Okay.
They can respond. They have odor detectors primarily around their lips, sort of, and a scientist showed that the main thing they're responding to is, I think it's sodium ions from salt and arginine, the amino acid arginine. I think those were the two. And that, if you just had water with those in it, that was pretty much the same as having blood in the water.
And then, so are they biting into the intestines?
Yeah, so I could show you a movie. What they suck on with they have both a front sucker and a back sucker, right? And I can show you a movie, actually. There's a video that they made from KQED. It's a program called The Deeper Look. So there's the back sucker, and that's the front sucker that they're using to explore with.
Okay.
And then the teeth are in that front sucker. So they like those. They'll go around and find a place where there's thin skin. These guys are moderately hungry, but sometimes they'll start biting within seconds. Actually, here's another smaller one. I should put that in there.
can, let's see, like that one there has, I don't know, they have a certain attitude that they get in place, and you can sometimes see them peristalsis sort of pumping to help pull the blood in. So there's a few of them. I'll put in some of these ones. You can imagine that they the blood is such a rich meal, and they take so much of it in if they're lucky, right? It's like winning the lottery, sort of. It's, you know, these would normally be in a stream or a pond, and they're waiting for deer or cattle or people, whatever, to come in. When they're babies, they'll eat frog blood. But as adults, they want more, so then they're really looking for mammals with blood, and it's sort of hard to find. They can't get to them very quickly, right? But if they manage to feed, latch on and feed, they'll take a lot, and then they can live for a year or more without feeding.
From just one feeding?
Yeah.
Oh wow.
Yeah. Yeah, and these, both the sausage and the blood were sort of old, so I think the sausage is starting to leak a little bit. But normally, no, and also they'll cut the hole, and then they'll say, "Oh, now I'm going to try over here," and then the blood starts leaking out from that place. But so they're just going to go on like that for a while, and then when it gets too gross and disgusting, I just pull them out and put them back in there. So that's how we feed the leeches. But like I said, this is the one we use the least in our research. The one we use primarily is these, and I don't know if there's any embryos I could show you. Oh. The red that you're seeing there, that's the gut of the leech, filled with, it looks like blood, but it's really just the lymph from these insect larvae that happen to be red. And then these leeches are all hermaphrodites. It's sort of curious. So they're simultaneously both male and female. I'm not seeing. And then this white material there, that's sort of the egg mass inside the parent, the prospective parent. Yeah, so this is pretty clear example of the eggs inside the parent. But then when they lay their eggs, they put them in cocoons on the ventral surface, and they make sort of a little pouch with by folding their body wall over them. I mean, these look simple, but they're really pretty complicated animals, and to think how they arise from a single cell it's an incredibly complicated process. I'm not finding any eggs.
So with them being hermaphrodite, how do they reproduce?
Yeah, so these actually, they have to cross-fertilize, so they mate with each other. But some of the others, species,
can actually self-fertilize.
Okay.
See the pumping action coming from that one.
Mm-hmm.
That one too.
Oh yes.
They spend about 10 days normally, and from the egg to something that starts to look like a leech. I could show you some. It's partly that my eyes are no good anymore. I'm going to see if there's anything on these, and then you can look. You have fun with the exam?
Wasn't too bad.
Good. And then what we do when we're doing the experiments is just hold the animal and with one pair of forceps and pluck off the embryos with the other, and then so I'll put these back. They're starting to be sort of sausage-like. So now he doesn't want to move much. He said, burp. Oh, can I just go sleep? Sometimes it can be sort of daunting and intimidating, and you feel like, geez, everybody else is smarter. You've got imposter syndrome or whatever. I guess an important thing to realize would be that pretty much everybody feels like that. You know, I feel like that, and I always felt like that when I was starting school too. So you just don't let those feelings get the better of you, and just take advantage of the opportunities that are there. And Bio 1A is not a very fun way to learn biology. The better way is to be in a lab doing stuff and learning stuff that's immediately relevant to your project, whether it's leech development or the molecular biology techniques that we use in the lab to study gene expression patterns, stuff like that. And then you're part of a small group, the lab group, where everybody has a shared purpose and shared interests.
Mm-hmm.
So that's the ideal. So if you're really interested in biology, biology is not what you learn in the class; it's what you do in the lab, right? And it's creating knowledge from scratch, discovering things for the first time that nobody ever knew before. That's what's really exciting. And so if you can find opportunities to do lab research, that's a good thing.